Hello everyone, I hope you're all having a great time. Today I would like to share with you an experiment I'll be conducting about photosynthesis. But before we get started, I would like to share with you and remind you of some information or things about photosynthesis that you might have forgotten. Photosynthesis is a chemical reaction. It's an anabolic reaction too because it fixes a smaller molecule, which is CO2, into a larger molecule, which is glucose. So it needs energy to fix a smaller molecule into a larger molecule. Now, as I said, it's a chemical reaction, which means that it has some indicators that prove that it's a chemical reaction. Some of those indicators is formation of precipitate, formation of gas bubbles, change in color, change in odor, and much more. We have three factors that affect the rate of photosynthesis. They are the light intensity, temperature, and concentration of carbon dioxide. In this experiment, I'll be conducting how the concentration of carbon dioxide affects the rate of photosynthesis. Now, in this experiment, we're going to need a light source, discs of organic spinach, which is 4 discs for each container. They have to be equal. Sticks, they can be of any type, a syringe, a stopwatch, 3 glasses with the same amount of water, which is 100 milliliter, and here we have the different amounts of bicarbonate of soda. Here it's 1 tablespoon, in the middle it's 2 tablespoons, and 3 tablespoons. And now we're going to start our experiment. At first, we're going to put the different amounts of bicarbonate of soda into the same amount of water, which is 100 milliliter. This is the 1 tablespoon. This is the 2 tablespoon. And lastly, this is the 3 tablespoon. And now we need to shake them really well. Now this is the container or the glass with the lowest concentration of bicarbonate of soda, which is 1 tablespoon. Here we have medium concentration, which is 2 tablespoons, and here we have the highest concentration, which is 3 tablespoons. And now we're going to try to put the discs, 4 discs for each container.
Yeah.
Now, as we see that those discs are floating in all of the different concentrations, here, and here, and here. This is because of the air sacs that the discs have. The discs have air sacs that are filled with gases. Those gases make the discs less denser or have less dense, which makes them float. So we need to make them sink. In order to make them sink, we're going to use the syringe. We're going to do the same process for all of the 3 different concentrations. We're going to put the discs inside the syringe after removing its cap.
This is the first desk. This is the second desk. This is the third desk. And this is the 4th desk.
Now, when we have those desks inside, we're going to try to put them in a little bit. And then we can use this one to tuck them in properly inside. Yes. Now we're going to add water, some 2 milliliter of water. In this way, we're doing vacuum. Vacuum, what happens in vacuum is that all of the desks, the air sacs inside the air sacs will be removed. It will be replaced by this solution, which makes it less dense, which makes it have more dense, and which makes it go down. So in order to make it sink, we're gonna do vacuum using the syringe. We're going to close from the top, and sometimes some syringe have a small hole. We can also close it. And we're going to press in and up, down and up, as much as we can, in this way, in order to try to remove all of the gases inside the sacs. So let's do it. We'll keep on doing this process until we see the desks coming down or sinking down.
We can do this.
Now we can see some of the desks are going down and some are still up. We can do the process again to make sure that all of them are going down and they have CO2 in their sacs. Now, as we can see, we have all of the desks down. So we're going to fill it back to the container again, and we're going to repeat the process for both of the rest containers. Now, as we can see, they're sinking down. Now I have my desks sinking down in all of the 3 containers. We know that light is one of the factors that affect the rate of photosynthesis, but it would be perfectly fine if it was in the same distance for the 3 different concentrations, because we are measuring the difference of concentration of CO2 affecting the rate of photosynthesis. So light should be constant for all of the 3. As those desks are exposed to the light, photosynthesis will take place in each container, which means O2 will be produced again because it's a product of photosynthesis. It will go again back to the air sacs that are on the desks, which will make them have less density and will make them flow again. So O2 is our proof that photosynthesis took place. Now, when we turn on the light, we will start our stopwatch to see how much time did it take for each container with different concentration to have all of its desks floating again. After exposing the 3 containers with the same distance to the light source, photosynthesis took place in each container, but at different rates because it had different concentrations of carbon dioxide. Now what happened is that O2 was released by the process of photosynthesis. It entered again to the air sacs. The entering of O2 in the air sacs made the desks have less density, so they were started floating again. Now I have recorded my experiment here. The highest concentration, which is of 3 tablespoons, took 1 hour and 35 seconds exactly. The medium concentration, which is of 2 tablespoons, took 1 hour, 10 minutes and 19 seconds. The lowest concentration, which has 1 tablespoon, took 1 hour, 23 minutes and 50 seconds. Coming to an end with our experiment, we concluded that the production of O2 is the main proof that photosynthesis took place. However, we also concluded that increasing the concentration of carbon dioxide or bicarbonate of soda when we have the same amount of water, light intensity, and water temperature, and the number of desks increases the rate of photosynthesis. Thank you so much. I hope you have a wonderful day.